Hello, I'm Mr. Baker. This is the biology content video for my unit on DNA and genetic information. So this video is going to talk about how new genetic information is formed, but we're just talking about new DNA here, all right? And when we say new DNA, we mean kind of new, right? Because when we make new cells through mitosis, we're going to need a new copy of all those chromosomes to go in it. So we're going to go through what's called DNA replication. And DNA replication is semi-conservative. That means each part of the DNA is going to continue to be used with new parts being added to it. So we've got this DNA ribbon here, right? This helicase, and it's going to be split. So we can see half of it goes down here and the other half goes up here. So these would be the first replication of this DNA strand. Each is 50% old and 50% new. If we go through another replication, we get 4 because 2 times 2 is 4. So this set, half is old, half is new, but this one is all new because the new version here got added to even more new stuff. All right, and same thing down here. So we call this semi-conservative. If it was completely conservative, it would just all be old stuff. If it's completely non-conservative, we would have old stuff and then a complete new stuff. So it would look like this and this. We just care about the semi-conservative part here. All right, DNA replication has 3 parts. We have initiation, elongation, and termination. Right? This is the basic breakdown. It does get a little bit more complicated as with most things in science. All right? So initiation is uncoiling the chromosome and the DNA helix and getting it ready for replication. Elongation matches new nucleotides to those existing structures and bringing the new nucleotides and existing nucleotides together and matching them up. Termination is recoiling that DNA helix and doing a little bit of error checking. Not as much is known about the termination steps as the elongation and initiation steps. We are mainly going to focus on initiation and elongation with only a little bit of termination. We're going to talk about error checking in a later video. So just take a step and breathe here. This picture does look complicated, I know. I'm going to pair this video for my class with a video as an animation that shows all this in motion, and the narrator does a little bit more work on going in depth than we need to worry about. All right, so we are going to talk about how once it is uncoiled and straightened out, so we're going to start here, right? Something called a protein or an enzyme specifically called helicase splits the nucleotides. All right, so it breaks the hydrogen bonds between adenine and thymine and cytosine and guanine. All right, so it breaks those bonds between the base pairs that you see in the corner opposite me, down there. Sorry, pointed the wrong direction. Okay, and the 3' and 5' sides of the DNA split. All right, DNA polymerase is the thing that is going to put it back together, and it works in the 5' to 3' direction. All right, so we're going to have a couple of different ways to talk about this. The leading strand and lagging strand. This image I stole off the internet is part of it is in another language, so ignore those. Okay, we'll call this a leading strand and this a lagging strand for our purposes. All right, so the leading strand replication happens away from the fork, and lagging is toward the fork. And this fork here is what we're talking about, how it splits, like the tines on a fork that you eat with. All right, so primase is going to come through and put an mRNA strand in place that works as kind of a spot for all this to start. So it kind of puts the first step in elongation here. And that is going to... Sorry, I had to cough and didn't want to record that. but I only do one take, so we're going to keep going. So the lagging strand is going to be from the 3' to 5'. Leading strand is 5' to 3'. And we're going to keep going here, right? So the lagging strand is going to be happening away from the fork, and the leading strand is toward the fork, right here. So this one is working this way, and this one's going this way. I know this arrow thing is kind of confusing. It's just showing that it's going to keep going away, all right? And so what's going to happen is primase comes in and sets it up. It puts a primer on. That's what this is called, the RNA primer. And it gives a spot for another enzyme to come in and bring these nucleotides to match up with the existing nucleotides. That's what I meant by existing and new, all right? The new nucleotides form what are called Okazaki fragments, all right? So they are kind of like long, I don't want to say strands here because these are already strands, long lines of matched base pairs that already have hydrogen bonds in them, right? They're starting to be hydrogen bonded to the old existing strand. And then DNA ligase is going to come in and remove the RNA primers, all right? So that's going to leave a little bit of gaps in this half of the strand, but they're going to be replaced as it goes, all right? So more and more groups of nucleotides are going to be present, and eventually DNA polymerase is going to join it all together, all right? So we've got primase putting a primer on, DNA ligase removing that primer, and then DNA polymerase joining the Okazaki fragments. They're just named after the guy who first discovered them, so his last name was Okazaki. These are called Okazaki fragments, right? This is going to continue happening throughout the process of DNA replication. And it's going to happen at multiple different sites in eukaryotic organisms as this fork here continues to spread and spread and spread until it gets to all of the DNA being split like this, all right? And when they're joined back together, they're going to have a leading strand or a lagging strand that is new nucleotides and a leading strand or lagging strand that is old nucleotides, all right? So again, we're basically doing an unwinding of those. That's called initiation. We're working on the base pairs, right? Elongation, and then joining them back together. That's called termination. All right, we're going to keep this simple. We're not going to overcomplicate this, okay? Remember your base pairs. This is A to T and C to G. I know when I put my cursor down there, it brings up the menu. So adenine to thymine, cytosine to guanine, and those go the other direction too, right? Thymine to adenine, guanine to cytosine. If you have any questions, make sure you reach out to me. We're going to continue working on this before we do anything else, I promise. So we're going to take this week, and then next week we're going to start looking at how this DNA gets used to do things in the form of proteins. So have a good day.